Proteins. They're what make up our bodies. They do most of the work on the inside of our cells, too. They control the contraction of muscles, they become enzymes to speed up chemical reactions, they're used in antibodies, they can be cell receptors, and so much more. As you probably know, people also need to consume proteins to stay healthy. People also consume proteins to gain muscle mass as well. In fact, people carry protein shakes in these to show the world, hey guys, I go to the gym. But where do these proteins come from? We call this protein synthesis, or protein biosynthesis. The steps of protein synthesis are described by the central dogma process, which sadly involves no dogs. Essentially, it goes from DNA to RNA to a protein. The steps from DNA to RNA is called transcription. RNA to a protein is called translation. As simple as it sounds, there's a lot more to these steps. Let's take a look at these steps individually. First, there's transcription, which occurs in the nucleus of a cell. It's important to know that transcription factors are what control the start of transcription. However, that's a whole other process that I shouldn't get into right now. An enzyme called RNA polymerase binds to the promoter region on the DNA. The sequence of this promoter region is TATA, or thymine adenine thymine adenine. This promoter region tells RNA polymerase where transcription starts and which strand of DNA is the template strand. The template strand is the template for the messenger RNA, which is what is built in transcription. mRNA will be complementary in bases to the template DNA strand, except it will have uracil instead of thymine. Next, RNA polymerase unwinds DNA bits at a time and moves along the template strand from 3' to 5'. RNA polymerase starts to build mRNA by adding complementary bases on its 3' end. Remember, the newly synthesized RNA must be anti-parallel to the template. At this point, the mRNA strand is actually premature mRNA. Post-transcriptional modifications must occur. In pre-mRNA, there are introns and exons. Introns are non-coding sequences, and exons are coding sequences. The introns must be spliced out via a spliceosome so the coding region of mRNA is not interrupted. Afterwards, a 5' GTP cap and a 3' poly A tail are added to the ends of mRNA. These protect the mRNA from enzymes in the cytoplasm. Translation is the last major step in protein biosynthesis. Now that mRNA is mature, it can go to a ribosome in the cytoplasm to be translated into a protein. Before we get into that, we must discuss codons, as they are what code mRNA into amino acids. A codon is a 3-letter sequence in the mRNA code that codes for a specific amino acid. The first codon that will code is AUG. AUG is always the start codon that initiates translation, but it also codes from a thionine. The rest of the codons will be read and translated as amino acids too, until a stop codon is read. UAA, UAG, and UGA are all stop codons. Specifically, how are these codons read and translated though? As mentioned before, the mRNA goes to a ribosome and it is pulled through. Here, transfer RNA, or tRNA, can move through the ribosome's A, P, and E sites, which are slots specifically for tRNA. tRNA has a 3-nucleotide sequence called an anticodon, and an amino acid connected to it. This anticodon complements a specific codon in the mRNA sequence. When the tRNA matches with its specific codon on the mRNA, the amino acid assembly starts to begin. This is why codons are important. They code for amino acids. As tRNA matches its anticodons to codons, the amino acids are linked together through a peptide bond, ultimately making a polypeptide, also known as a protein. The ribosome is the structure that helps create this bond between the amino acids. This is why the mRNA goes to the ribosome in the first place. So, to sum it all up, RNA polymerase moves along a template strand on the DNA and builds mRNA in the nucleus. Post-transcriptional modifications happen to mRNA, and mRNA goes to the ribosome. In the ribosome, tRNAs read the mRNA, matches its codon to the mRNA's codons, and amino acids are assembled to build a protein. That's it. Thanks for watching. See you next time.